All right, so here's our lecture for tomorrow, and it's going to cover the last portion of Chapter 7. It's also going to present to you questions from Chapters 6 and 7 that we're going to spend time in class tomorrow discussing, and it will present a paper that we'll also spend some of our time tomorrow discussing. So, sex-linked disease. All right, key terms, just two of them. First one, oncogenetics. This is something we were planning on getting to yesterday in class. We didn't quite get there, and it's basically the genetic process of cancer formation. And you'll see in the next slide, we'll go through the different mutations and how they are inherited, and basically the progression, the genetic progression of cancer formation, of forming cancerous cells. Immunogenetics, this is basically the genetic process of basically training aspects of your immune system, the genetic process of building the adaptive immunity. And we're going to focus just on antibody and T cell receptor formation, and talk about how we can generate millions of different versions of these antibodies and T cell receptors with just a few genetic pieces. All right, so oncogenesis. So tumorigenesis basically goes in this progression. First, you get what's called a growth signal autonomy. So that is the cells no longer require a growth signal in order to move from G0 into the cell cycle. And that could be triggered by a number of factors. It could be triggered by the overexpression of a cyclin-dependent kinase, or an overexpression of something that deactivates an inhibitor of a cyclin-dependent kinase. It could be overexpression of one of our cyclins. It could, and oftentimes is, an overexpression of certain cell receptors that basically trigger processes, trigger transduction pathways, even if they're not binding a particular ligand. So that's the first step. Basically, growth signal autonomy, the ability to move through the cell cycle without a growth signal. The next one is unlimited replicative potential. So the average cell in our body can replicate, I think it's about between 50 and 70 times before it reaches senescence. Since the telomeres shorten with every replication, after that number of replications, you've shortened the telomeres enough that you are losing chromosomal information, and eventually the cells just can't survive anymore. Now, we've talked previously, this is erased in germ cells because of the work of telomerase to lengthen the telomeres. Now, in cancer cells, the unlimited replicative potential can be accomplished by either an overexpression of telomerase in cells that would normally not express it at all, and so it keeps those telomeres nice and long and allows the cell to replicate probably close to an unlimited number of times before it reaches senescence. Or it can be accomplished by what we call non-homologous recombination, and there are machines that will use that to lengthen the end of chromosomes. All right, evasion of growth inhibitory signals. So there are signals in the extracellular environment inhibiting growth, and so many cancer cells at this point will downregulate the expression of certain receptors that will bind these inhibitory signals, or it could also downregulate the expression of some of the proteins that work in the transduction pathway after those inhibitory signals are bound to a receptor. Evasion of apoptosis. So P53 is the product that basically as a result of DNA damage will lead to the upregulation of several different inhibitory molecules that will stop the cell process until the DNA can be repaired. It also can trigger the apoptotic pathway, and so this is oftentimes accomplished by the downregulation or the elimination altogether of P53. Angiogenesis is a process where the cells signal the vessels nearby to grow additional pipes, if you will, to provide additional blood flow to the growing tumor. This tends to happen later in the process. And then finally, invasion and metastasis. And this is basically the last stage when the cells lose their ability to remain attached to other cells and to the extracellular matrix, at which point they can invade surrounding tissues and get into the bloodstream and move around the body. All right, so we need to talk about specific mutations. Mutations in proto-oncogenes, and if you'll remember, proto-oncogenes are genes that code for cyclins, genes that code for materials that might inhibit these cyclin-dependent kinases, or code for materials that will destroy the inhibitors of cyclin-dependent kinases. And so mutations in these have a dominant inheritance pattern. One mutant copy is enough to get the cell to just progress through the cell cycle without growth factors, without growth signals, and without any real care for any errors. And so these have a dominant inheritance pattern. A single dominant allele is enough to trigger the shift from a normal cell to a cancer cell. Mutations in tumor suppressor genes, and these are genes for, say, the inhibitors of cyclin-dependent kinases, genes for, machinery in the transduction pathway for receptors that bind inhibitory signals. These have a recessive inheritance pattern. So as long as you have one normal allele for these tumor suppressor genes, you typically will not develop cancer. That's enough to keep cell growth in check. And so these mutations tend to follow what's called Knudsen's hypothesis, it requires two mutations in order to develop cancer if the cancer is involved with this tumor suppressor genes. And his hypothesis is basically that some individuals or cells inherit one mutation. So some individuals may inherit that mutation from one of the gametes that fuse to form them, or even a specific cell might inherit one of those mutations after a round of mitosis. And then the second mutation comes as typically the result of a mutagen, or it could come as the result of another copying error later down the line. But that second mutation tends to come later and then eliminates the ability of this tumor suppressor gene from functioning properly and ultimately leading to cancer. Other one, mutations in mutator genes. These also have a recessive pattern, and these mutator genes are like the genes for DNA repair machinery, or the genes for some of the other machinery that will check for DNA damage, those kinds of genes. And those also have a recessive pattern because one normal copy is typically enough to ensure that the cell fixes DNA damage properly, that the cell checks for DNA damage properly, and if necessary, inhibits cell growth. All right, so immunogenetics. Here we've got basically the one thing we're going to talk about is the formation of antibodies and T cell receptors. Basically, we've got three separate genes: the D gene, the J gene, and so the V gene, the D gene, and the J gene. And so we draw these as though they're right next to each other. It's not really the way that they look inside of the human genome. They aren't right next to each other. However, they do function together. And these genes are only active in cells associated with adaptive immunity, B cells and T cells. And so what happens is in each of these three genes, we have alternative splicing. So we've got the V gene that can be cut in various ways, that various parts can be cut out as introns during the RNA maturation process. We have the G gene or the D gene that is the same. It can be spliced in various ways and have different portions cut out as introns, and the same for the J gene. And then after all of these are transcribed, they are basically put back together. They are the transcripts are put back together either prior to translation or after translation when we put these proteins together. But most often this is going to happen prior to translation to create basically a single transcript that is a combination of the mRNA from 3 separate genes that all of them can be cut in alternative ways. And so you've got your variable domains that can be cut in various ways, and then we have all of them have this constant domain to ensure that all of these things hold together. Antibodies and T cell receptors are a combination of these factors of heavy chains and light chains, and a variable region where you basically have on the section that binds whatever that foreign material is. And this allows the body to make literally millions of different antibodies and T cell receptors from just 3 genes that can each of those 3 be spliced in alternative ways. Now, there are 2 categories of antibodies specifically. There are monoclonal antibodies that bind one specific material, and then there are what do we call them? Multiclonal or polyclonal antibodies that combine multiple different materials in kind of the same category. And so what we're going to do is we're going to talk a little bit about a paper that we're going to spend a good amount of time tomorrow talking about, that basically talks about a sex-linked genetic disease and how it can be treated by designing specific antibodies. And so it's going to pull in some of this material as well. All right, so the disease is hypophosphatemia. Hypophosphatemia is an X-linked dominant genetic illness. So having one mutant copy of that gene is enough to trigger the genetic illness. So this is a genetic illness that's going to be more common in females than it is in males, because females have 2 chances to get the one mutant allele and males only have 1 chance to get the one mutant allele. And so it's going to be about twice as common in females as it is in males. And basically it leads to an overproduction of fibroblast growth factor 23, abbreviated FGF23. And what this does is basically it leads to hypophosphatemia, because this fibroblast growth factor 23, FGF23, makes the kidneys have a very difficult time reabsorbing both vitamin D and phosphate. Now, vitamin D is essential in having proper bone growth and formation, and so this leads to rickets. Having very low levels of vitamin D and very low levels of phosphate in the serum leads to bone deformities and ultimately leads to rickets. Now, most form of rickets can be treated extremely well by just giving patients excess vitamin D. This one, it doesn't work that way because giving them excess vitamin D doesn't keep that vitamin D serum levels high, because the kidneys are still not reabsorbing any of it, and it very quickly drops back down to lower levels. So the research team in this paper treated hypophosphatemia with monoclonal antibodies against FGF23. And so they basically, in order to build these custom monoclonal antibodies, if you will, you present a B cell with this mutant form of growth factor, and you allow the antibodies to you present these to B cells, and whichever B cell has an antibody that can bind this then replicates like crazy, producing a lot of antibodies against that, and then now you can take those antibodies and you can treat patients with it. And when they did, they saw an increase in both serum phosphate and vitamin D levels. The kidneys became more efficient at reabsorbing those materials, and there was a reduction in the pain associated with rickets. They were able to demonstrate very clearly a reduction in the pathology, and a lot of it had to do with allowing the kidneys to function properly in their reabsorption of phosphate and vitamin D. So again, we'll spend some more time tomorrow talking through this paper, but here are the questions that I want you to be prepared to discuss tomorrow. The first three are from Chapter 6, the latter three are from Chapter 7. What is the role of RNAs in cytoplasmic inheritance? And so we talked a lot about cytoplasmic inheritance and that it comes from the mom. But it's a lot of that is these RNAs that are already present in the cytoplasm. Well, what do they do? What role do they play in regulating gene expression? What is the supposed role of skewed X inactivation in the development of autoimmune diseases? What are the differences amongst the symptoms of a carrier woman if the X-linked gene encodes a soluble product, or if it encodes a cell-bound product? How would those two conditions differ in how a heterozygous woman would demonstrate the phenotype? What are iPS cells? What is the role of SRY and RSPO1? And then finally explain the Knudsen's hypothesis. Okay, so be prepared to answer these questions, be prepared to discuss this paper, and if you have any questions on oncogenetics or immunogenetics, you can certainly ask those tomorrow.